So thank you so much, Steve, for that really wonderful introduction. And it's a great honour for me to be here to share this special day with you, and also to express my admiration for the development of your Brown Institute for Neuroscience or Brain Science. So today's topic, I think, is relevant particularly to psychiatry, but also to other brain disorders. And it's really relevant to the general issue about psychiatric classification. Should we be satisfied with a categorical system, or should we allow dimensional features to interact with our understanding of mental health disorders? And I'm going to focus on two dimensions, two behavioural dimensions, if you will, in this talk, to see what kind of utility they may have for understanding certain brain disorders. So these dimensions, one of them is a highly popular one, impulsivity, noted with many citations in the Web of Science here. It's an intense area of study in psychology and brain science and genetics even. And compulsivity, which is perhaps a more modern invention, hardly studied, but undoubtedly important, as you'll see, for many mental health disorders. Now, this diagram is supposed to demonstrate the fact that clearly there is some kind of overlap between these two, because both of these dimensions imply some kind of disinhibition for impulsive behavior or compulsive behavior. Maybe inhibition is something they share, but in psychological terms, they almost certainly comprise different components, different mechanisms, which I'm not going to go into great detail in this talk on. And also, intriguingly, they're associated with a different pattern, perhaps, of mental health disorders. But let's perhaps first, before we describe those, let's consider some working definitions. So, impulsivity, actions which are poorly conceived, prematurely expressed, they occur too soon. Unduly risky, inappropriate to the situation, although impulsivity may sometimes be beneficial, often it's not, and may often result in undesirable circumstances. By contrast, compulsivity, similar definition, but here we're talking about actions which persist inappropriate to the goal. They occur over and over again in maladaptive situations with no obvious relationship to the goal, and again, often resulting in undesirable consequences. So the prototypical disorders, if you will, of impulsivity will obviously include ADHD, or attention deficit hyperactivity disorder, which impulsive behavior is one diagnostic symptom. But also they're going to include mania, certain personality disorders, the dysregulation syndrome in Parkinson's disease, and also, very importantly, substance abuse and eating disorders. Compulsivity, well, intriguingly, as I'm going to be arguing, the recent diagnostic manual emphasises no fewer than about 10 symptoms in substance use disorders, which can all be rebadged as compulsivity, failures of cognitive control, eating disorders, but also prototypically OCD, and I will be talking a bit about OCD towards the end of this talk, also influencing some other conditions such as autism. Now, one way of conceptualizing these dimensions, as I'm going to argue, is that impulsivity often occurs in anticipation of rewards, at the beginning of a response sequence, before you've selected an option and decision, whereas compulsive behavior often follows from an outcome of a response sequence, what you do next. And so that's a rough differentiation of these two psychologically. In neural terms, we might imagine that they're associated with these celebrated frontostriatal pathways. So on the right here, you see the human reward system with its head ganglion in the orbitofrontal cortex and the ventral stratum, or the nucleus accumbens, with inputs from the limbic system, the amygdala and the hippocampus, and so on. And this motif is repeated throughout the frontal lobe sector to include also motor functions, as in Parkinson's disease, but also more complex functions, barely understood, but involving cognitive processing. Moreover, these functions, as we know, are under powerful chemical modulation, particularly the monoamines, dopamine, noradrenaline and serotonin, which mediate the fluctuating circumstances of arousal, stress, mood, etc. And so I'm going to be arguing that impulsivity and compulsivity actually engage different circuits in these frontal lobe circuits. If we twist them on their side for the moment so that the red circuit is now at the top, I'm going to be arguing that impulsivity in particular involves ventral striatal substrates with prefrontal connections, and compulsivity involves more dorsal striatal elements associated even with elements of the motor association cortex and premotor cortex. Now, I mentioned earlier the human reward system, and I think we have to recognize that most of the inspiration for the brilliant imaging studies on the human reward system have been inspired by studies in animals. In particular, studies trying to model human drug addiction. And here we have one of the arguably most successful models in the whole of human neuropsychiatry, that of drug self-administration. So in this procedure, rats or mice or even monkeys may self-administer, they choose to self-administer small solutions containing the drug of abuse through an intravenous catheter into the vein. And this behavior actually is predictive of human drug abuse potential and is used as an assay for such. I think the only drug that humans perhaps abuse that rodents don't is LSD. They don't like LSD very much. But then you could argue that humans don't really binge on LSD either, so it may be a special kind of drug. But in this elemental study, the late Bartley Herbal of Princeton arranged it so that rats didn't self-administer amphetamine, the dopamine-releasing agent, into the vein, but directly into the brain through implanted cannulae, targeting different structures, but eventually settling on the hot spot in the basal forebrain in this structure, the nucleus accumbens. And our own work with Barry Everett picked up on some of this a decade later by showing that not only would animals self-administer amphetamine to this structure rather selectively, suggesting reward center or reinforcement center, but they actually did so according to optimize the level of dopamine there, because in this study we replicated Herbal's results, but also infused dopamine blocking drugs, dopamine receptor blockers, and demonstrated that rats would adjust their self-administration to achieve an optimal level of dopamine in this structure. Dopamine clearly innervates the accumbens from its important ventral tegmental projections in the midbrain, and the fact that other receptors, notably nicotine and opioid receptors, are in the vicinity of these dopamine neurons suggested a common pathway, a positive reinforcement pathway for drugs of abuse. And I think this concept has very much prevailed until today at least, and moreover it's been translated. And some of the most powerful translation has been by the brilliant work of one of your previous speakers, Nora Volkow of NIDA, who showed that in human brain of drug abusers, whether alcoholics or methamphetamine abusers, was a downregulation of striatal D2 receptors, shown here, as compared to the healthy normal brain. But what I wanted to highlight is a couple of other aspects of her study here. First of all, a remarkable correlation or relationship with a reduction of D2 receptors being correlated with a hypoperfusion on brain imaging of the orbitofrontal cortex, thus showing in one glimpse the activity, if you will, of the frontostriatal circuit relevant to drug abuse. Now that's also important because, as you know from the work of Damasio and others, that damage to this kind of area produces really devastating impairments in decision making. So then you begin to hypothesize about addiction. Do some of these potentially neurotoxic effects of drugs of abuse on structures like the orbitofrontal cortex, by impairing decision making and inducing risky behavior, actually exacerbate the drive to addiction? It's quite a possibility. Once it starts, it gathers momentum. The other issue is this, and it's not one you immediately think, but on first perception of a reduction of D2 receptors, one's thought is, this looks like an adaptation in the brain to very high levels of dopamine-releasing drugs. In other words, an effect of the drug. But maybe it's not that. Maybe that, and indeed this change, occurred before drug exposure happens. In other words, these changes may be present in the individual before they become drug abusers. Now, how do you test that in humans? You can't test neonates with PET and follow them longitudinally to see if from birth or during early life, possibly as a consequence of stress, they show a D2 downregulation, which leads them to addiction. It's a very hard study to do. Perhaps you need some animal studies. And quite by chance, Jeff Daly and I discovered something which may be relevant to human stimulant addiction, which depends on a paradigm we invented for rats. This is modelling a human task called the continuous performance test. In this situation where rats, or mice, it's been shown in mice, monitor a range of apertures to detect visual targets which are associated with food delivery. If the rat correctly nose pokes in the appropriate aperture, then it's rewarded with a pellet, and then the next trial begins for 100 trials. So this is a continuous task. Now, I'm not going to go into details on this task, except to say that if the rat gets it wrong, it's punished by being plunged into darkness for a few seconds and failing to get a food pellet. He has to start the next trial in a humiliating fashion with an empty magazine flat. But if he also responds prematurely, in other words, tries to guess where the computer's going to put that visual target, is also punished. So premature responding is bad. And yet, some rats in the Lister hooded strain, about 10 or 11% of them, this one's name is Zippy, persistently show this tendency to impulsive behaviour, which loses them a lot of food pellets. They detect the visual events okay once they give themselves a chance to do so, but they persistently show impulsive behaviour. Now, this is almost certainly a trait because it breeds true. We've done the behavioural genetics. We haven't published it quite yet, but the changes are in chromosome 1, and they're associated with the GABA receptor B, also the mGluR5 receptor, and a range of synaptic proteins. We'll come back to that later. But the intriguing thing about these rats is that although their phenotype is pretty normal on the whole, they're not hyperactive, for example, put them in the presence of cocaine with an intravenous catheter, and they over-respond in this binge access procedure. So the green are the high impulsives, the red are the normal rats. Some of them have to be restrained from administering an overdose. So this is very intriguing because we have here rats who are impulsive before they ever saw the drug, and now they're showing increased drug-related behaviour. Now, as an aside, I should also say that the drug actually makes them less impulsive, and this is reminiscent of the effects of Ritalin in ADHD. And that's another talk, but I won't come back to it. But what I will also say is that these rats have another interesting phenotype, which is low D2 receptors in the ventral stratum. So using microPET, Jeff Daly was able to show this effect, which predicted the degree of impulsive behaviour they had. And this study's been replicated now 3 times. So these high impulsive rats also, before they ever see cocaine, have low D2 receptors, just suggesting the possibility that in humans there might be a similar relationship. We further analysed this low downregulation of D2 receptors with autoradiographic measures of the D2 receptor and also the dopamine transporter in the nucleus accumbens core and shell regions. Now, you know the nucleus accumbens is divided into these 2 areas, and the main changes to the dopamine markers are in the shell region. So that's where there's a reduction of D2 receptors in the high impulsive rats, and this is also where there's a reduction of dopamine transporters. So, together with some neurochemical data we've collected and others, we hypothesized that there is an upregulation of dopamine in the shell region of the accumbens. And moreover, when amphetamine is injected there, it induces impulsive responding. Now, Together with this, there are also changes in the core region, intriguingly, the other region of the accumbens. And this has been highlighted by a recent study by Caprioli and Jeff Daly and we in Cambridge, showing a reduction in voxel-based morphometric measures of grey matter, if you like, in the core region, which again beautifully correlate, certainly on the left-hand side, with the degree of impulsivity. This is a very high correlation coefficient. So again, the less, if you like, grey matter in the core, the more impulsive the rats are. Moreover, if you have molecular markers, the GABA decarboxylase, which of course is a marker for medium spiny neurons, which are in predominance in this region, that's reduced in the high impulsives, certainly on the left-hand side. And some synaptic proteins, notably sphenophylin and MAP2, are also downregulated. Just to put a cap on it, if one puts antisense for GAD into the nucleus accumbens core in normal rats, you get an increase in premature responding. So we think that in the accumbens, there's an interaction between the core and the shell. Actually, it's a competition between these two structures, because if damaged, they have opposite effects on impulsive responding produced by amphetamine. And this has been part of an enterprise by which we've tried in a very primitive way, without the modern neuroscience tools so far, to define the neural network which controls impulsive responding in the rat of this type. We call it weighting impulsivity, because it also extends to temporal discounting of rewards. And this circuitry includes not only top-down circuitry, including the infolimbic cortex, importantly, which regulates the shell region, and the cingulate cortex, as well as the hippocampus and the insula, which isn't on this graph, but also these bottom-up chemical systems, not only dopamine, but also serotonin and noradrenaline. I don't have time to describe our work on the neuromodulation of this system, but it is complex, and we have quite a lot of evidence. But the big question really is this relevant for humans? So this notion of high impulsivity contributing to stimulant drug abuse and addiction, is there any substance to it? Well, this is work with Karen Urscher we've done to look at stimulant abusers, mainly cocaine and amphetamine. This is a typical study where we use age and IQ matched control subjects. And this is the task, among other tasks, we've used to quantify impulsivity in human amphetamine addicts. We have an animal version of this as well, by the way. This is Logan's stop task, familiar to those of you who study cognitive control, whereby the computer directs a directional response to two buttons, left or right, very rapidly. But occasionally a beep sounds, and on that sound you must inhibit your response. So just as you're pressing that down button, the beep comes on, and you have to stop it. By modelling the response time distributions, you can obtain a measure not only of go reaction, but also of how long it takes you to stop. And an inverse measure, that is, of impulsivity. So the longer the stop time, the more impulsive you are. And this task has been validated by we and others in patients with adult ADHD, for example. And it responds to Ritalin, by the way. But in these studies, where we applied it to drug abuse, we were inspired, first of all, by a set of studies by Adam Aron, when he was a student with Barbara Sarkin and myself, looking at frontal patients, damaged brains with all sorts of injuries to frontal lobes, measured carefully with magnetic resonance imaging, and then quantifying regions of interest precisely into several regions of interest. For example, here the right inferior frontal cortex, medial frontal, and so forth. And what we found was a striking correlation, highly significant, between stop signal reaction time and the volume of damage to this right inferior frontal area. It's the only area which showed any semblance of correlation. And it was not correlated with the go reaction time. And we know subsequently from imaging studies that this is part of a network which is involved in the breaking of behaviour, the stopping of responding. Karen Urscher applied this paradigm to groups of stimulant-dependent individuals, as well as healthy controls, but also to other groups of interest. The first group were the siblings of the chronic drug abusers, the siblings who do not take drugs, never have done. This is what we call an endophenotype study to see whether there might be a semblance of impulsivity in the family. Okay, that's the concept. And the other group of interest are recreational stimulant abusers, and I'm sure you have some of these in the States. We have them particularly in the city in London. These are young high flyers who take a lot of cocaine at the weekend. Often very smart. They never quite deserve a diagnosis of stimulant drug dependence, so we call them recreational stimulant abusers. Now, on the stop task, you can see some dramatic differences. First of all, the chronic dependent users are very impaired, as you'd predict, but so too are their siblings. They've never taken drugs, or hardly ever, and yet they're as bad almost as their drug-abusing partner siblings. The recreational drug users aren't at all impulsive. Intriguing. Not only that, but there's a neural correlate of this deficit, which can be measured with fractional anisotropy of white matter projecting to the frontal lobe here. So this particular tract, the arcuate fasciculus, the density of this is related to performance on the stop task, both in siblings and in the chronic drug users. So the implication is that a weakening of connectivity of this maybe inferior frontal area is prodromal to vulnerability for stimulant drug abuse. Now, whether you want to say this area is involved in willpower and that really drug addiction is a matter of overcoming willpower is another discussion, but it's an intriguing neural correlate of this inability, potentially. Now we move to compulsivity. I've been inspired throughout my career by the work of Chuck O'Brien at the University of Pennsylvania. He's one of the doyens of clinical psychopharmacology. And this quote from The Lancet, for me, sums it up. At some point after continued repetition of voluntary drug taking, the drug user loses the voluntary ability to control its use. At that point, the drug misuser becomes drug addicted, and there is a compulsive, often overwhelming, involuntary aspect to continuing drug use and to relapse after a period of abstinence. Compulsivity. As I said earlier, now the dominant symptom characteristic for substance use disorders, dominant over withdrawal and tolerance now. This is how you diagnose it. Can you measure compulsivity in animals? Well, remember our definition, persistence in the face of adversity. So, in our paradigm, sometimes we give rats electric foot shock for self-ministering cocaine. And this considerably reduces the enthusiasm of normal rats for the drug. As you can see in this graph, this is the very low levels of self-ministration in what we call low impulsive rats. But the high impulsive rats, defined as earlier, continue to self-administer at a very high rate, even though their pain thresholds are normal. So this is what we define as compulsive drug taking, drug seeking, from impulsivity to compulsivity. And there are other measures as well of persistence which also single out these individual rats as resistance to punishment, in this case, as potentially drug-addicted rats. This corresponds to the human situation, whereas we saw before, mere exposure to cocaine and stimulant drugs isn't enough to make you addicted. Only about 16 to 20% of such individuals merit that diagnosis on DSM. So there's individual variation in response to these drugs. And this led Barry Everett and I to a controversial hypothesis, which has a neural correlate. So impulsivity, mediated, as we've seen, by the ventral striatum, by the nucleus accumbens, this reward prediction system, may, with chronicity, with repeated drug taking, devolve into compulsive behaviour, which hypothetically involves progressively an involvement of the dorsal striatum. So as behaviour becomes more compulsive, and we're going to argue habit-based, so other regions of the striatum are recruited to mediate this behaviour. So this is the primate basal ganglia, if you will, or striatum. Here's the shell region, the core, the putamen, the caudate. And there is a cascading circuitry which connects these areas in a rather vectorial manner. It only goes one way, from the shell to the putamen. So this is how the shell activity here ultimately can influence processing in the putamen, which we are going to argue is particularly involved in stimulus response habit learning as a building block for compulsion. So in parallel with this devolution, hypothetically, during the progression of drug use to addiction, of course, we would also argue there is a progressive loss of top-down control from the frontal cortex. So you need both of these elements to give you addictive behavior. Now, I'm not going to give you all the evidence that we've and others have arrayed in order to support this controversial hypothesis, but I'll just show you one snapshot of one study which shows the kind of thing that we mean. So this is a study of Luc van der Schoen in our lab showing in the self-administration procedure that if you block dopamine receptors in the dorsal striatum, early on in learning there's no effect, but later on in learning you get a progressive deficit, a reduction in drug-seeking behavior, which is quite interesting. What's even more interesting is that if at this stage you also infuse the drug into the nucleus accumbens core, there's no effect. Now, I'm not saying the nucleus accumbens has lost all forms of control over behavior at that stage, but clearly it's much less important after extended training than initially. We know this structure is essential for the acquisition of this behavior early on. So there's been a change, and there's other data in the literature which is consistent with this viewpoint. Now we're going to switch a little bit to talk about compulsivity in a broader context before I describe some of its underlying behavioral and neural mechanisms. So first of all, let me describe this study we did a few years ago using then the unusual technique of resting state connectivity. Imagine that you're in the scanner with your eyes open, lying back, thinking of nothing in particular. In this study, which was done with a 3 Tesla magnet, we were able to compute the interactions between 50 regions of interest in the human cortex, cerebral cortex. And by looking at the intercorrelation matrix of the activity in those regions, devise a proxy measure, if you will, of their connectivity. That's the very simple-minded idea. The degree of correlation of activity in these regions must presumably reflect, in some sense, at least their functional connectivity, if not their structural connectivity. And so, in this study, we included not only healthy controls, but also a group of stimulant drug abusers and patients with OCD, obsessive-compulsive disorder, who prototypically show compulsive behaviors. And to our amazement, we found that in these 2 different groups of patients, there were 2 areas only which were relatively disconnected with respect to the rest of the cortex. They're both in the frontal lobes, one's in the frontal pole, and the other's here in the orbitofrontal ventral medial prefrontal cortex. There's the quantification of the connectivity. And even more intriguingly, this connectivity was related psychiatrically to their performance on psychiatric scales of compulsive behavior. Notably the Ogden scale for compulsive drug use, which asks questions like, you do drugs, doing drugs is interesting compared to liking drugs, for example. And the YBOC scale, the Yale scale, of course, is a measure of clinical OCD. In both cases, there's a striking correlation, negative correlation between the degree of connectivity and the level of compulsivity, suggesting, of course, some common construct, controlled possibly by some overlapping circuitry in these 2 very different psychiatric presentations, compulsive drug use and compulsive behavior. Well, it's interesting if you look at patients with substance use disorders, stimulant drug disorders, not only are they more impulsive, but they are more compulsive as well on the YBOC scale. So they show higher YBOC scores than control subjects, and by the way, so do their siblings. So they do show our stimulant drug abusers some signs of OCD, which is interesting. There's some overlap. Which leads us to the question of, well, what is compulsivity? That's the definition I gave you earlier, the persistence. Here are some possible mechanisms. We can study this at the level of stereotypy, which is the repetition of small acts, often displacement behaviours in zoo animals, for example, or in response to high doses of amphetamine in humans, behavioural stereotypy, or enhanced habit learning, which habit learning is an automatic form of behaviour which is separate from goal-directed learning and has a degree of automaticity. Reversal learning, where one has flexibly to adjust behaviour and a decision task to adapt to the change in rules in the task, or extinction, where reward is omitted altogether. Persistence in either of these could be taken as an index of persistent compulsive behaviour. Rigid cognitive set, attentional fixations also may contribute to substance abuse, eating disorders, gambling, or OCD. So let's consider two of these in the rest of the talk, two of these mechanisms. The first is this analysis of instrumental behaviour, which I referred to earlier as self-administration in the case of drugs, where we can redefine this as action-outcome learning. In the presence of a particular environment or stimulus, this is a rewarded environment and leads to a reward or a reinforcer. And that association is associated with a particular response. This is the mechanism for instrumental learning, often called now action-outcome learning. Learning the consequences of your actions. It's goal-directed behaviour. Versus something which probably goes on in parallel with goal-directed behaviour. Maybe it's a more primitive system, we don't really know. Stimulus-response habit learning. So this is the direct evocation of a response by a stimulus. You're in this environment, what do? You do that. What do you do rather than what are the consequences of your action? This is the distinction being made. How do you diagnose these interwoven strands of processing which govern all of our behavioural outputs? All of our behavioural output is a mixture of goal-directed behaviour and habits, clearly. Well, two ways are to devalue the goal, for example, by poisoning food, or by simply providing satiety for a particular flavour so that you no longer value that food. Satiety is a very good devaluing agent. And observing whether the behaviour continues unabated. If it does, under devaluation, presumably it's not dependent on the goal. And therefore, by subtractive inference, it is habit-based. That is the assumption. The other way of diagnosing a habit is to uncouple the action from the outcome subtly. And if you degrade the instrumental contingency and yet behaviour persists, again, habits are diagnosed. I'm just going to describe devaluation experiments in this talk. Now, some very clever scientists, Tony Dickinson is one of the leaders of the field from Cambridge originally, John O'Doherty, have taken this to the brain by using actually food, very often, and showing these systems in the brain engage in different circuitries. So in one fMRI series of studies, the goal-directed system, of course, includes this ventral medial prefrontal cortex area and the caudate nucleus, actually, for goal-directed behaviour, whereas, again, using devaluation procedures, habit learning seems to invoke more dorsal striatal mechanisms, including the putamen and probably the premotor cortex. So there's neural evidence for separating these two. How can we make use of this in studies of mental health disorders, particularly addiction and OCD? I'll try and describe some attempts for that. First of all, here's some work of structural brain imaging from Karen Urscher in our group, looking at the brain structures of individuals in the groups we described before, the stimulant-dependent individuals, the unaffected siblings, and the recreational drug users. So these are horizontal scans, and the blue coding means reduced grey volume, on grey matter volume on the basis of structural scanning, and the yellow and hot colours mean increased. What you see in these chronic drug abusers is notable reductions of grey matter volume in a variety of cerebral cortical areas, particularly frontal and also some temporal lobe areas as well. Now, these changes are not necessarily present, as you see in the unaffected siblings. So only limited changes that I described earlier are present in the unaffected siblings. And presumably some of these reductions in grey matter are caused by drug exposure. And indeed the evidence for this is that they get worse with increasing duration or chronicity of drug exposure. But the other interesting observations are that there's increased grey matter in the basal ganglia, including the putamen, and this is also even present in the unaffected siblings. So there's a hint that maybe these subcortical systems, including the habit system, are upregulated in these drug-abusing groups, but not in the recreational drug users. As you see, if anything, they have more grey matter than normal, perhaps because they're very smart enough to protect them from the ravages of cocaine exposure. Behaviourally, how does one get from a habit to a compulsion? This is something which I should raise, perhaps it's an item for discussion, because clearly compulsions are much more than habits. But there are three mechanisms that we can consider. First of these I've already mentioned, the notion of impaired top-down control. If you have a habit which is out of control, then maybe it becomes a compulsion. That's the hypothesis. Another aspect is sensitization. So we know from the work of Terry Robinson and others that repeated drug exposure actually makes you more habitual. And the evidence for this comes from work in rats, where the balance between goal-directed behaviour and habitual behaviour is swung in favour of habit behaviour by a chronic regimen of amphetamine exposure in rats. Not self-administration, but just giving rats amphetamine. Just makes them more habitual. It's a very interesting result. And it's shared with the effects of stress. So stress often cross-sensitizes with amphetamine itself. Stress itself swings the balance in favour of habit-based processing away from goal-directed processing. So all of these factors may work together to convert adaptive habits to maladaptive compulsions. So the present focus is on OCD, which often involve obsessions, sometimes very weird ones, and anxiety, leading to compulsions, repeated behaviour. This is the traditional account of OCD, what I call the linear account, the linear causal account. Obsessions evoke anxiety. You reduce the anxiety by performing a behaviour which reduces, and then it doesn't go away, actually. It continues. So that's the OCD cycle. Maybe hand washing, maybe check-in, all sorts of bizarre compulsions. But another way of conceptualizing it from our perspective, particularly given our interests in translations from animals to humans, is to focus on compulsions, to focus on behavior, which you can observe in both. And this hypothesis that habits may contribute to compulsions can be studied in animals and humans. So, compulsions. I think it's worth focusing on them a bit because of a number of interesting considerations for OCD. Compulsions can, after all, generate anxiety as well as alleviate it. Convulsions can include mental acts, thinking compulsions. They can occur in the absence of obsessions, whereas obsessions, or pure obsessions, occur only rarely. Consider the enigma of compulsive hoarding, which often occurs without OCD symptoms, anxiety. Consider that response prevention is the dominant cognitive therapy in OCD, rather than mere cognitive exposure. Consider the fact that anti-anxiety agents in general don't work in OCD, notably the benzodiazepines. And also consider the fact that DSM has agreed to separate OCD from the anxiety disorders. So although anxiety is an important part of OCD, I'm interested in these compulsive aspects, which I think make it shared with other compulsive syndromes, including maybe gambling, anorexia, eating disorders, maybe even drug addiction. So our own OCD patients, again, this is a typical group. We do select them so that they don't have clinical depression. They may have higher scores on depression scales, but they are not comorbid with clinical depression. They clearly will have anxiety. And we select them regardless of their particular compulsion, whether it's hoarding, ordering, checking, obsessing, or washing. And we've studied goal-directed and habit learning in this group, and indeed in subjects with substance dependence, in three main paradigms of habit learning. One, a computational paradigm borrowed from Nathaniel Dorr. One, an appetitive goal-directed habit paradigm called the Fabulous Fruit Game, and another aversive habit learning, and this isn't a game. So first of all, considering the computational paradigm, I'm not going to describe this in great detail, it's quite complicated, and my ex-student Claire Gillen, working with Nathaniel Dorr at NYU, has done, I think, a lovely study with this. This is a complex two-stage decision-making task where basically, if I can simplify it, the real aim of the task is to get the big picture of what's going on in this task, that sometimes you may have to make decisions in the short term which are disadvantageous in order to reap a greater harvest in the longer term. If I put it like that, you have to really understand what's going on in the task. And we call that model-based learning. It can be computed. Versus responding to the immediate contingencies. What happens on this trial? Were you rewarded or punished? Were you successful or not? What does that make you do? Does that make you win stay or lose shift, for example? And this can be modelled in reinforcement learning terms as model-free learning. And you can measure the relative mixture of these two forms of learning in the task with a so-called Y parameter or X parameter, which gives you the ratio of model-based versus model-free processing that an individual or a group is exhibiting. And what Claire was able to show is that this task, when you use this measure of compulsivity, is predictive of, well, about a dozen questionnaires which are factor analysed to produce three main factors: anxiety, depression, social withdrawal, and compulsive behaviour. And the performance on this task, this simple task on this web-based Amazon Turk population of healthy individuals, collects a huge proportion of the total variance of this factor of compulsivity. So the degree to which they use the model-free strategy rather than the model-based strategy, so this is negative from model-based towards model-free, that is predictive of all of these questionnaires gathered on the web. So that's a striking result, suggesting you can measure objectively some of these dimensions with psychological tests. And why we think that's interesting is that if you use this ratio, so low ratios scores meaning increased dependence on model-free or what we think is habitual-based processing versus model-based or goal-directed, more goal-directed processing, you can see that in binge eating disorder compared to healthy volunteers, or OCD, or stimulant drug dependence, in each case there is a greater tendency towards the model-free, we think more habit-based processing. Not in obese subjects per se, as a kind of control. So that's one line of evidence. Another line of evidence is with a simple psychological task, the Fabulous Fruit Game or the Fabulous Animal Game. So this is a test of associative learning, whereby you have to learn associations between fruits as stimuli and fruits as goals or outcomes. So the fruits as stimuli are on the outside of the box, and in the presence of this banana cue, you have to remember or learn whether to press the left or the right button in order to earn a reward fruit, a cherry, which is worth 5 points. Okay? You've got to learn several of these at once, so there's quite a lot of learning going on here. And you'll recognise this is classic instrumental learning: stimulus, action, outcome. And actually, OCD patients learn this fairly normally, intriguingly. But you can devalue the outcomes, as you can in the animal experiment, simply by saying, well, this one no longer earns points. It may even earn penalty points. So for heaven's sake, don't earn those fruits because they're bad for you. So you do a devaluation test. And then you give them a quiz, or you do a behavioural test, or you do another test of habit learning by very rapidly presenting the stimulus fruits one after another and seeing whether the subject will go for it, in other words, make a response to earn the outcome or not. So clearly for some fruit stimuli, it should inhibit because the fruits have now been devalued, where others they should go. So it's like a no-go procedure. And what happens under this situation is that the OCD patients continue to respond automatically to the stimuli which were formerly associated with valued fruits but have now been devalued. So they're habit-based. They're showing what we call slips of action. And amazingly, I think, this correlates very highly with the Yale-Brown Y-box score for symptomatology. So this is an appetitive task. There's no real punishment here other than losing points, which doesn't count for much. And yet you can predict, to some degree, their OCD. By contrast, if you ask them about consequences of their actions, they're very bad. They're very bad whether you ask them which button do you press when that fruit's on to get that fruit, or when you actually give them a behavioural test between two fruits, do you press left or right? And the OCD patients just don't remember which are the consequences of their actions. So in the words of at least one of them, they learn what to do rather than what the consequences of their actions are. So they're habit-bound in operational terms. Now, we've done a similar experiment recently in stimulant drug abusers, or actually they have some opioid abuse as well. This is the fabulous animals task, but it's actually exactly the same as the other one. I won't tell you why we had to use animals for drug abusers. But intriguingly, in this task, they're actually impaired even in learning, the cocaine abusers. But on these tests, both by questionnaire and behaviourally, they are bad at predicting the consequences of their actions. They have impairments of goal-directed learning, but they have increased slips of action in the go-no-go test. Now, this isn't simply because they're disinhibited and impulsive, because we have a control test for that, where we actually tell them to not respond to certain stimuli as a kind of equated measure. And they're vaguely impaired on that, but this impairment of executive control does not predict their impairment or their increase in habit-based responding. So by these criteria, stimulant drug abusers share some of the habit-bound imbalance that patients with OCD are also exhibiting. The third paradigm is avoidance. So in this task, very simple task for humans, done, I should say, with full informed consent and ethics, involving electric shock presented to the wrists of subjects to a level which is mildly painful but tolerable, but certainly aversive. In the presence of the blue stimulus, you're going to get a shock to the right wrist unless you press the right foot pedal. If you get a yellow stimulus, you're going to get a shock to the left wrist unless you press the left pedal. In the presence of the red stimulus, you're safe. That's safe. Subjects learn this task very quickly, indeed. Very quickly, in very few trials. And that's the foot pedal part. Now what we're going to do is devalue. And the devaluation is it's an instructed evaluation. It's simplistic in its naivety, if you like. We simply disconnect one of the shockers from the wrist in full view of the subject. So the electrodes are no longer there. And the subject's quizzed on this quite intensely. You can't be shocked on this wrist, right? There's no electrode there anymore. Do you agree? Yes. No, I'm quite safe. It's fine. It's excellent. But you can be shocked on the other one. Okay. So one of the shocks is devalued, if you like, and the other one retains its value. Your only task is to avoid the remaining shock. And then what we do is test the subjects after a small amount of training and after prolonged training. And you only get these effects that I'm going to describe after relatively extended training. What happens is this. For the valued shock, there is no difference. And there's never any difference with GSR measures or subjective measures of anxiety or any of that between the OCD patients and the controls. But we find rather remarkably, and we've replicated this on a number of occasions, they do continue to respond for the devalued shock. In other words, they tend to continue to make the response which used to avoid a shock, but now no longer has to avoid a shock. And the patients make up all sorts of reasons why they're doing this, including the fact, rather irrationally, well, I felt I could still get a shock somehow, even though they've just agreed that they can't. Intriguing. So, operationally at least, this is a habit. Okay, and it's correlated, intriguingly subjectively, with what we call a premonitory urge. So subjectively, but again, when you ask them, they say, I felt I had to do this. I had an urge to do this. It's an urge. So we've imaged the urge, okay? We've done a functional imaging study of this paradigm. We've replicated the behavior results in the scanner. And we showed, I'm going to have to omit some of these data, a hyperactivity during learning in the ventral medial prefrontal cortex, as you would predict, Steve. But the second part of the experiment I think is more interesting. We divided the patients, there are no controls now, sorry about that, into two groups. One group showing this anomalous habit-based behavior, which is ridiculous, but is shown by over half the subjects. The other half don't show it. But that does give us an opportunity to look at the brains of these two subgroups to see what's special about the habit-based behavior. And what's special is that there is an activation in the basal ganglia in particular, in the habit-based group. Here it is. It's actually more in the caudate nucleus than the putamen. So that's against our prediction, I should say. But it's in the basal ganglia, these two subgroups. The premonitory urge is associated with a hyperactivity in the caudate nucleus. And it's correlated with the premonitory urge. What's even more interesting is when you do the so-called psychophysiological interaction analysis to analyse what other parts of the network are active or inactive for that activated site in the caudate nucleus, we find the following. Two areas of interest in particular. One area, the inferior frontal gyrus. Remember this? The inferior frontal cortex on the right-hand side, involved in impulsive top-down control, inhibition, braking, the stop signal task. This is underactive when this blob is active in the habit-bound OCD patients. So they have less active top-down control over that caudate focus. And in the subgenual cingulate cortex here, they have increased activity, which is partly part of this residual ventral medial prefrontal activity, which I mentioned occurs during learning. And for those of you who are rodent experts, you'll know that this structure has a very important role in toggling control, switching control from goal-based behaviour to habit-based behaviour. And Anne Graybill, but also Simon Kilcross, has shown this in a rodent study, whereby quite remarkably, if you damage the rodent homologue of this area, which is the infolimbic cortex, you can turn a habit-based rat into a goal-directed rat, which is fascinating. So the goal-directed behaviour is still there, but it's latent. It's masked, overwhelmed by the habit-based behaviour. But you can change the balance with this executive toggle switch. And that's what I think is so interesting about this study, showing how the habit, maybe in a nascent form, in a preliminary form in this aversive context, is under control by top-down mechanisms in the frontal cortex. So in the last few minutes, and I think I have a few more minutes, it started reasonably late, I want to discuss his other dimension, we think of compulsive behaviour, more cognitive dimension, rigid thinking or strategy, often studied in humans with the Wisconsin card sort test, and I hope for this audience I don't have to describe that in great detail. But I will anyway. So this is a test used by clinical neuropsychologists, often for diagnosing frontal brain damage, because of Brenda Milner's original discovery. It's a test where you have to sort cards according to trial and error feedback and learn a rule and then shift the rule according to that feedback. So you may initially have to sort cards by colour, but then the rule changes and you're told incorrect after a correct sort, and you have to think, what do I have to do now? Learn the new rule. Frontal patients can learn the rules, but they perseverate in the old rules, sometimes even realising that they're doing it wrong. Remarkable. Similar ego dystonia to what you may see in OCD, perhaps. In our animal-based version, which of course we also use to test humans, we have just 2 dimensions, shapes and lines, and we use a touch-sensitive screen because marmoset monkeys have great problems picking up cards. They don't have a thumb. They throw them up in the air and do unmentionable things to the cards. So we use a touch-sensitive screen. They have to find which predictive icon, whether it's a shape or a line, best predicts banana juice. These icons can occur in all combinations with one another, in any place on the screen. So you have to zero on which is the best predictor. Having found that, we may change the exemplars to see whether you've learned the rule that shape is the thing to focus on rather than lines. That's called an intradimensional shift. An extradimensional shift is when you change the examples, but now, for the first time, you have to shift from studying shapes to lines, thus showing cognitive flexibility of rule learning. And this is analogous, of course, to the shift in the Wisconsin test. The other form of flexibility is what we call affective shifting, where the stimuli stay the same, as you see here, but their emotional valence is switched, so that what was positive is now punished and vice versa. That's called reversal learning. And we discovered 20 years ago that these 2 different forms of cognitive flexibility depend on different prefrontal circuits in the primate brain. We showed this with excitotoxic, that's fiber-sparing cell-body lesions of the orbitofrontal cortex in the marmoset, which is probably areas 11, 13, 14, and the lateral frontal cortex, areas 12, 47. And here you can see the double dissociation. Reversal learning impaired by orbital lesions, as had been found by Butters and others in rhesus monkey. No effect, however, on rule shift learning. Quite dramatic result that compared to normal animals. Whereas the lateral lesion impaired the rule shifting but not reversal learning. We've replicated the results in a number of ways, including looking at their striatal outflow aspects and also their neurochemical concomitants, because in a number of studies, which I don't have time to describe in detail, carried out by Hannah Clark with Angela Roberts and myself, is to show that serotonergic manipulation, particularly of the orbitofrontal cortex, selectively impairs reversal learning without affecting rule shift learning, extradimensional learning. It's a remarkable neurochemical selective effect. Whereas manipulation of the catecholamines, notably noradrenaline and dopamine, affect rule reversal or rule shifting rather than the effective reversal learning. So there's a kind of double dissociation at the neurochemical level, which, who knows, might result from the fact there's more serotonin transporters in the ventral frontal cortex than in the dorsal frontal cortex. Intriguing. Moreover, this also happens in the rat. So in rats, we've tested trait compulsivity, if you like, in the rat in terms of their tendency to perseverate in a simple spatial reversal task. So here, you reward going to the left and punish going to the right. Now you switch it. For how long will they persist going to the left? And there's enormous individual variation in this. Some rats persist an enormous amount and others switch quickly. When you look at these different quartiles in this rat population, you see a reduction in serotonin transmission both in levels of 5-HT and the metabolite, and in 5-HT2A binding in the orbitofrontal cortex, particularly in the lateral orbital frontal cortex. So low serotonin, poor reversal learning, and increased perseveration as an index of compulsivity. Now, why is that interesting for OCD? OCD is treated with very high dose SSRIs. Nobody understands why. It's not because necessarily they're depressed. We think it has some definite effect on compulsive behavior mediated by the orbitofrontal cortex. That's a hypothesis that we want to continue to test. And indeed, in our rats, we find that high perseveration is remediated by the selective serotonin reuptake blocker, citalopram, but not by atomoxetine, which, by the way, is a splendid treatment for impulsivity in rats and in humans and in ADHD. So here's a kind of neurochemical dissociation as well between aspects of compulsive behavior and impulsive behavior. So now taking this again to OCD, this paradigm, using a houses and faces task, which is entirely analogous to the shapes and lines paradigm, Adrian Owen and Adam Hampshire have shown, using the BOLD technique, that there are indeed dissociable networks mediating extradimensional shift learning, which tends to activate the ventrolateral prefrontal cortex, as you'd predict from the marmoset work, and reversal learning, which also activates parietal cortex and lateral orbital frontal cortex, as here. So, as in the marmoset in human, we see with imaging a similar dissociation between the two forms of cognitive flexibility. And we also see this in rats and mice when you use touch and smell as the modalities rather than aspects of visual stimuli. But anyway, in OCD, patients and their relatives, given this faces, houses task, show relative underactivation of the lateral orbitofrontal cortex compared to control. So here's the lateral OFC activation, normals, relatives and OCD patients, and the network change also in parietal cortex, posterior parietal cortex. So again, this is a kind of endophenotype for OCD, this propensity to not perform the reversal task normally, let's put it that way. What about rule shifting? Well, behaviourally, we find rule shifting, it's such a strong effect. This is the so-called Cantab version of the IDED shift task, where you shift from shapes to lines eventually. We find that patients with OCD and their relatives are very bad at learning the ED shift. So they learn the rule normally, but on the ED shift, compared to controls, they take more trials and more errors to reach the learning criterion. So again, these subjects are a bit rigid in their behaviour, and we think this may contribute as an element of top-down control deficit to the development of compulsive behaviour. This is true both for medicated and unmedicated patients, as we would have predicted on the basis of our rat and marmoset work, that serotonin doesn't do very much for this kind of higher-order rule-shifting requirement. So this is work of a new student, Mathilde Vagui, at Cambridge. And what Mathilde's also done is use this resting state, lying in the scanner with your eyes open paradigm, to get measures, proxy measures of functional connectivity, this time between the frontal cortex and the stratum. And she's related this to performance of the shifting task carried out outside the scanner. So, quite remarkably, these are all of the OCD patients that we've tested individually. And their performance is a function of the underactivity of this circuit, including the, well, BA1047, this is relatively frontal pole, ventrolateral prefrontal cortex, and the head of the caudate nucleus here. So this is, this shifting impairment is linked to an underactivity of the lateral frontal caudate loop in these patients. So we've seen both underactivity of lateral circuits and relative overactivity of medial circuits in OCD. And the question then arises, how can we possibly take advantage of this for therapeutics? One possible approach may be, and of course this centre would know this possibly better than anywhere else in the world, deep brain stimulation. So we've recently been obtaining permission to do the first study of deep brain stimulation in OCD in the UK on 6 very severely affected OCD patients. These are patients who are imprisoned in their own homes because of their compulsions. And this is a study of Eileen Joyce with the neurosurgeons at Queen Square and a whole team of workers, including we doing some of the neuropsychology. The unusual aspect of the study is the targeting of 2 different areas in the same patient with different placements. One focused on the ventral capsule overlapping with the nucleus accumbens, and the other focusing on the well-known focus for Parkinson's disease, the subthalamic nucleus, which has also been used in this centre and others to treat OCD. So I'm just going to show you some preliminary data from these 6 subjects, which in our consideration are remarkable. So here's the clinical data. So the Y-box score, well, you'll know is an index of OCD. These are very high Y-box scores. And they also depressed, these patients as well, on the depression scale. And this is the effect of SDN stimulation. There's quite wide variation, but overall the trends are clear that with both sites of stimulation, the anterior capsule of the SDN, or with both combined optimally in the same patient, there is a highly significant reduction of the symptoms. So at least one of these patients subsequently obtained a partner and went on holiday to Barcelona for the first time for 20 years. This is incredible. These are patients that didn't respond to drugs or cognitive therapy. One of them did not respond, I should say. And one of them, intriguingly, has shifted away from OCD to stimulant drug abuse. An interesting observation. But I'm just going to show you this one snapshot of our cognitive battery, the ED shift task, which really is improved. This is the average baseline of all the patients. And this is the effects of the stimulation. You see, the SDN site produces a remarkable improvement in flexibility, whereas the accumbens or ventral capsule site has huge variance and has no significant effect. So I think this is intriguing because we can begin to show possibly different elements of OCD in these different circuits contributing, and we may have to seek different remediation of these circuits to produce a completely beneficial effect, if you like, on OCD. Okay, so in summary, I've been talking about an analysis, if you like, of OCD symptoms and some associated conditions, which involve 3 main categories: an imbalance in the habit and goal-directed system, which we think also is implicated in the progression to addiction, failures in top-down control, as exemplified notably by shifting in OCD and perhaps by response inhibition in drug addiction, and probably emotional factors, emotional vulnerability to stress, to anxiety, which, as we've seen, promote the drift to habit-based behaviours. So I've described evidence which shows a circuitry involving impulsivity in rat, and indeed we have the evidence in humans now as well, which I haven't described, implicating ventral stratum and certain areas of prefrontal cortex in impulsive behaviours. Shifting and inflexibility, one of these intermediate loops involving the caudate nucleus, and potentially the habit-based loop involving the putamen and motor association cortex. So my collaborators include Barbara Sahakian, Barry Everett, notably on the drug addiction studies. Ed Bullmore supervises our work with functional brain imaging and structural brain imaging. Angela Roberts runs our non-human primate lab. Mathilde Vaghi contributed data. Karen Urscher leads our substance abuse work. And Jeff Daly, of course, works on these neurobehavioral concomitants of impulsive behavior in rodents. Thank you very much. That's a really interesting question. We had a discussion this morning where we were talking about habits becoming valued for their own sake. So habits becoming more rewarding than instrumental actions in OCD, for example. So I don't see in principle why that shouldn't be the case for impulsive behaviour, but I've never thought of it before, so that's an interesting suggestion.
Very, very nice talk. Many people recover from their addictions, and I know you and others have shown that there is some cognitive improvement with recovery.
Yeah.
But are you finding that the pattern of improvement is similar to the way they got, the reverse of the way they got there, with the reduction in the compulsivity and the brain circuitry reflects that? Is there any evidence of that?
It's a brilliant question, and it's one we should try and look at. I mean, you're right, we have studied in part what happens to abstinent drug abusers and whether there are residual impairments. And there are some residual impairments, which is one clue, and some of those residual impairments are in decision-making cognition, so, you know, that fits. But we haven't yet done an analysis which shows the unmasking of goal-directed behaviour in the way that you're suggesting that may happen in a recapitulated sequence in terms of its development. I think it's an interesting idea. Thank you.
Wonderful talk. I have a question. You showed the unaffected sibling data, unaffected sibling data for substance abuse, and that's, you can understand that because if they don't start abusing, then they might have the same biomarkers, but they haven't started abusing drugs. But you also showed unaffected siblings for OCD, and so I'm wondering why they have this similar biomarkers but don't have the disorder.
Oh, right. Okay, well, I think probably a question I often get is, okay, these siblings who are siblings of substance-dependent abusers, they've got really bad inhibition, stop signal reaction time. Why aren't they taking drugs if it's so important? It's a really good question. Normally, an endophenotype should be intermediate. We do see some measures which suggest that they aren't quite as bad as their stimulant drug partner. So that's part of the answer. In other words, they don't have a big enough dose to tip them over into addiction. But I think there are other factors, and I think one of them is resilience. And we have got some data which bear on that, using the STOP paradigm in chronic substance abusers, their siblings, and healthy controls. When they're adult, the substance abusers have underactivity of this inferior frontal cortex region in the STOP task. Their siblings have overactivity relevant to, compared to controls, overactivity. So it's as though they're exerting more effort in order to resist the temptations of drug abuse, if you like. We also see this overactivity in a sample of adolescents, 14 years old, who are being screened for drug taking. Those who are beginning to take stimulant drugs and other illicit drugs also show this increased activity at this area. But if they develop into long-term abusers, clearly this activity collapses and they become underactive. So there's some kind of effort, if you like, being imposed to control the behaviour, which then hypothetically collapses as the addiction progresses. That's the way we would see it. You know, another possible mechanism is simply different stress or trauma for these individuals, that you just need, a stressful episode in one sibling rather than the other to topple one into addiction and the other is still safe. So we don't know whether the family basing of this, of course, is genetic or family-based. It could be all to do with stress. One of the important factors we've found recently in drug abuse is simply being subjected to abuse when a child. And so, these kind of stress factors are also probably determining the expression of the compulsive drug-seeking behaviours.
And you would say that's probably true for the OCD?
Well, by analogy, I would say it's the same kind of argument, yeah. Yeah.
Thank you.